Good evening, everyone. Uh, I think we can start the session and then post the session. We can have another 10 minutes of discussion. And again, I'm sorry that I was not able to take any class on the weekdays. I had been busy, but I'll try to compensate. Maybe tomorrow I'll try to have one more class. I'll put the details today, and mostly we will be completing this general course. And from tomorrow onwards, we will go to the literature search and all. So just yeah, so I'll just first take you briefly just through the slides which we have discussed earlier regarding formulation development. Uh, so uh, regulatory framework and FDA requirement, we have discussed this. Most important thing is the formulation development, having a B equivalent study, then the B equivalent statistical criteria, and quality standards we have discussed. Then we have, of course, discussed the ICH guidelines, Q1, Q2, Q8 to Q12 guidelines. These are the important guidelines into consideration. Then Q1A guideline related to stability testing, Q2A, Q2R1 related to process validation, Q8 R2, which is the most important guideline for a formulation scientist, that is for pharmaceutical development. Q9 quality risk management, it is important for the R&D as well as the production and QA people. And again, Q210 is more important with the quality people, but again, important for R&D people. Then we have discussed pre-formulation, formulation, process development, stability, and analytics. So we have taken a very brief discussion about this, and as we move forward, we will discuss this each and everything in detail, with mostly with case-specific examples, or I'll be building up a dummy project. In fact, I have taken a case of albendazole tablet, and I have that presentation ready with me. So the same presentation we'll take, and then we will discuss everything in detail. API characterizations, we have discussed this. The UV HPLC data, FTRIR data, NMR data, then the DSC and TGA is required. Particle size, of course, is the most important requirement. Then XRPD is required. Flow properties again becomes a very important property, specifically when you are manufacturing tablets. Then the stability and purity of the API, usually governed by the force degradation data. And the impurity profiling, again, a very important aspect, because whatever impurity comes from the API, it will be reflected in your product. Then the physicochemical properties of the API. So it includes solubility, stability, and physical properties. We have discussed this in details. And then different type of analytical method selection, which parameters we include for analytical method selection and API selection. That also we have discussed in detail. Excipient selection strategy, we discussed this also. For functional performance, quality attributes, and cost optimization are the three important thing. When we say functional performance, it includes all the parameters which will have impact on your CQS. Then the quality attributes about the API, and again, of course, it has to be a cost compatible or cost effective excipient. Compatibility study we discussed, and again we will be discussing in detail with example. Then formulation development approach also we discuss, how much time each process can take, and in case of tablet, which are the process by which we go, what are advantages and disadvantages of the product we have discussed. So what we will be doing, we will be uh, uh, first taking one example from tablet. Then once tablet is done, we will take one example of injection, then we will go for solute suspension, then we will go for the topical preparations. Then we will again come back to tablet, and in tablet, then we will take examples of one for entry code and one for suspend release code. So in this way, all the dosage form will be covered. So and also what will happen when we take different example, we will be doing same activity but for a different dosage form. So there will be a thorough revision for each of the working of the general working, how we go ahead with the project. So it will be like a revision and might be useful for you. Quality by design. So earlier we used to do by quality by testing. Now quality by design is important. We have already started implemented. We started only for the US market, but now QBD is the approach for each and every market. Uh, Many market are right now not asking for it, but still we are going ahead with it, and that is what everyone in the industry is doing. Analytical method also we discussed earlier: development, optimization, validation, and method transfer. Out of this, method transfer is something very important. Method transfer I will discuss in detail. There are different type of method transfer: direct transfer, indirect transfer. All these things we will be discussing when we complete the QBD section of dosage form. We will go into QBD section of analytical validation. There we will discuss again with an example. Method validation requirement. Again, during the QBD of analytical method development, we will be discussing this also in detail. Then the ICH guidelines. Am I audible? Because I'm getting a message that the Zoom meeting is on hold. Uh, can anyone let me know whether I'm audible or not? Hello?
Yes, sir, it is audible.
It is audible. Okay, okay. Okay, so these guidelines and different zone stability study required and all, this we will again discuss with examples of the project from tomorrow when we start. So this will be something very detailed zone requirement, and then we will discuss how we can design a common stability study for multiple market. I have planned a full session for it, but this will come towards the end. So process development, process design, optimization, and control strategy. These also we discuss. And then when we complete the QBD section, I have plans to discuss this design of experiments with proper examples, and then design of experiments. We will do it online on a software, Minitab software. We will do it, and we will make it a more interactive session because this is going to be something very important. But DOE we will go only once we complete the general QBD part. We will not jump directly into DOE, otherwise it will be jumbling for you. So scale up considerations during scale up, what all is required. A very brief discussion we have done, but again, there will be a full-fledged session dedicated to this. Technology transfer protocol, this also we will discuss. We have discussed already, we will discuss in detail. Bioequivalence study, in fact, for this bioequivalence study, once we complete the QBD part, we will have a very full-fledged session dedicated only for bioequivalence study. Maybe on one Saturday or Sunday, 2 to 3 hours, we will just discuss with it, and I will come up with some actual examples from my earlier experience and all. I'm yet to make a presentation for it, but I will dedicate at least 2 to 3 hours because this is something most important for any formulation development scientist who is working in generics. So we completed this, and then now we come till B study we have completed. Now this is somewhat a regulatory part where ANDA preparation and submission. So right now I'm taking it only for the US market, but moving forward we will discuss different type of dossiers, how dossier will differ from US market to UK market to Europe market or ROW market. So we will discuss this one by one when we go ahead. Right now for ANDA, so as I told you, ANDA is nothing but abbreviated new drug application. So whenever we say ANDA, it is meant only for the US market. That is why it is called as ANDA. So ANDA framework, there are some requirements. So the core requirement for any ANDA is there should be an equivalence. So the API should be same. You cannot have a different API and then you can apply for a ANDA. It is not possible. Dosage should be same. Means if it is tablet, you can make a tablet only. You cannot make a capsule or some other dosage form in a routine ANDA. And if at all you change the dosage, then it will not be a ANDA. It will be actually a 505(b)(2) filing. So the 505(b)(2) filing is different than ANDA. When you change the dosage or some other thing, it is usually called as A. But when we talk about ANDA, ANDA is not about ANDA is only about having the same API, same dosage, same strength and bioequivalence study. So strength also needs to be same. It is not that innovator is 50 mg and you say I will make a 45 mg drug and I will do. There is a possibility that means if strength is the scope of A and A, there is a possibility that you can change the strength, but at that time you will not be an A and A. You will be either a 505(b)(2) application or some other application depending on the market. I'll just give you one example. There is a soft gelatin capsule of diclofenac. I think it is by the name of Xanodine capsule. And I am not able to remember. Xanodyne is the name of the organization. I will just write it down in the LinkedIn. So there is a case where I think Xanodyne is Xanodyne is a company and they made a liquid soft gelatin capsules by the name of Zipsor. So in this case, what they have done, they have claimed and they have proved that their 25 mg soft gelatin capsule is equivalent to 50 mg of normal diclofenac tablet. They have done a BE study and they have proved the BE equivalency, and then they have come as a 505(b)(2) application. So when you change the strength or the dosage and all at that time, it doesn't come in the scope of ANDA, but it is mostly a 505(b)(2) application. In rough sense, we can call it as a paper ANDA also. So this is a requirement for ANDA. While submitting, you have to have a same API, same dosage, same strength, and bioequivalence study. Quality, of course, wherever in whichever facility you are working, in that facility, you need to have a CGMP compliant manufacturing and controls. Labeling, it is in any A&D, it is always a compulsion to have a labeling similar to RLD, exactly like RLD. You cannot have your own labeling. So the normal timeline for preparation is 12 to 18 months for the development. This is something general which has come. Uh, in my organization, we consider that if a product is a IR product, we develop it in 12 months, and if a product is sustained release, 18 to 24 months. And FDA review, so usually FDA review takes 10 months for a standard cycle, but again, it depends upon the situation. Sometimes it goes to 2 years also, sometimes it comes in 8 to 10 months also. Then there can be number of queries. If the FDA is not satisfied with queries, you will get a full-fledged query, and sometimes you get minor query which you have to reply within a period of 30 days and all. So if there are multiple queries, sometimes this review cycle takes 2 years, 3 years also for approval. Uh, then the strategic approach. So pre-ANDA. So usually pre-ANDA is mostly meant for the injectable products and for the NCE. For usually for ANDA, because the US FDA has given so much clear guidance and guidelines. A control correspondence is not required with FDA, or a pre-ANDA meeting is not required in case of solid dosage forms. A pre-NDA meeting is mostly required sometimes for the injection product, where you have to give your proposed formulation to the FDA. You have to communicate that this is going to be my qualitative and quantitative formulation. So you give your approval. That's why a pre-ANDA is required. And many a times for NCEs, because my organization is very much involved into NCEs, so we have been doing a lot of pre-ANDA meetings. Exactly, it is not a pre-ANDA meeting; it is a correspondence meeting, which we proactively inform the US FDA about it. And then certain amount of time when you have a uh, what you can say, if you have, even if you are working on solid and all, and if you are stuck up and you are not clear, it is always better to have a control correspondence with the US FDA. So this is the case with the US FDA. With UK, that is MHRA and EMA. What happens that the guidelines are not clear, and also usually it is always better to have a pre-ANDA. It is not a pre-ANDA meeting, it is just a control correspondence with MHRA or EMEA, because for US FDA you can get every guideline with a proper year. For example, if you are working on an ANDA project, you clearly know what is going to be your RLD, which strength is going to be your reference strength and all directly from the website. This is not possible in the case of UK market, that is MHRA and EMEA. So it is there is some general guidelines of selecting and picking up the innovator, but it is always advisable to communicate with MHRA and ask them that this we are taking as a uh, RLD strength, we are going to do bioavailability against this strength, and these strengths are being considered as a biowaiver strength. It is always better to communicate in case of UK and EMEA. In fact, I have a very classical example where we were developing levothyroxine tablets. A levothyroxine tablet has multiple strengths. So, in this multiple strengths of levothyroxine tablets, strength starts right from 12.5 mcg and it goes to 300 mcg. So, we were developing a common formulation for both US market and the Europe market. So we had a very clear picture of our bio strength in case of US market. And also what we did, we went ahead with the bio study for the RLD strength mentioned in the orange book uh, for levothyroxine. And then we did a common bio study for both US and Europe. So what happened? Because US it was mentioned clearly, so our strategy got accepted. But then Europe people, they came back to us and they said that we have a different opinion. You cannot take this strength for a backbalance study. You should have a different strength. At that time, we thought that we should have communicated earlier. It would have saved a lot of time, and then we have to rebuild the project. Means US FDA it went ahead, but for Europe we have to again rebuild a project, do a bio study with another strength we have to give. So it is always better to have a control correspondence in case of UK projects. QBD guideline, of course, this is a strategic approach, and for US FDA, it is must to follow Q8, Q9, Q10 during development, because they see the development. But in case of UK, MHRA and EMEA, it is not required right now. But if you are not doing it, there is so many of queries coming. So it is always better that follow this guideline for the UK and MHRA also. And ECTD, you might be knowing, it is the electronic submission. So any documentation package and mostly specifically for the US market, you require module 1, module 2, module 3, and module 5. So module 1 is usually the administrative information, like the forms, patent, and the labeling which you have to give. For module 2, it is usually the quality and clinical overviews. Module 3 mainly consists of the drug substance and the product information, and module 5 usually consists of the clinical study report. So what we will do when we go with example, I will build up a dummy because of course, we cannot take any live project from the organization and all. But I have dummy folders what is required, and in each folder I will make a dummy document by the name, so that you people can understand which documents are required in the submission. So I'll have to work for it, but I will definitely work once we complete the literature review part and all. We will discuss in detail the parts of in various modules, module 1, 2, 3, 5, what information is required that we will be putting, and we will be discussing it. So anyone who intends to go into regulatory and all, it will be helpful for them. Then the post approval and launch changes. This again we will study in detail when we go there. So process controls and validation. So whenever we submit an ANDA and all, and based on the FDA 2011 guideline and the ICH Q8 and Q11 guidelines, uh, a FDA process validation framework needs to be submitted, and it is a three-stage approach where you have to submit your process design as stage one, process qualification as stage two, and continued verification as a stage 3. This is a very integrated slide which I have prepared. Very short and crisp slide I have tried to cover in just one slide. But moving forward, we will have a very full-fledged discussion because this is going to be important activity, and this is something very important for formulation scientists also, but also for the scientists who are involved in technology transfer and material science and technology. So we will discuss this in detail at that stage, and this is again important for anyone who is working in production also. And process control strategy also we will discuss. Right now, you should know that this is a requirement. So validation stages, there are again design, qualification, verification. So design is nothing but defining manufacturing process and the control strategy. You have to always define your manufacturing process with a very proper control strategy. What is your process and how you are going to control it? Then you have to qualify. So when we say qualification, so qualification, your facility needs to be qualified, your equipment needs to be qualified, and your batches needs to be qualified. And when we say verification, so ongoing monitoring to ensure process remains under control is verification. So maybe uh, only thing is that I should be able to get sufficient time. So as I told that in case of bioequivalence, we will have a 2 to 3 hour session post completion of the QBD approach. So what I will be doing for this process controls and validation, I will be making a very detailed presentation. And if you people, all you people have time and all, no, then we will spend at least any of the either Saturdays or Sundays or a mutually agreed time. We will have to spend 4 to 5 hours in a continuous discussion. We cannot have discussion today and have discussion some other day. So those discussion I will try once I'm thoroughly ready, and the day I think I have a full-fledged time, either on Saturday and Sunday, we will discuss in a letter, because this is again a very, very, very important aspect which everyone should know. So control implementation, validation stage, and control implementation. Of course, you have to give this document earlier, not required, but right now you have to identify and control your CPPs without the validation ranges. You have to write it on a document, and this document needs to be present in the facility. In process testing, release testing, environmental testing, all these things are required. In process testing, release testing, you have to have a proper uh, specification and all, and we will again discuss a session of how to set the specification, what are the targets for setting specification. We will discuss this in detail moving forward. Then process control strategy we have discussed, control philosophy, control hierarchy, and technology integration. Yeah, that is a very important aspect of process analytical technique. This is again very important aspect. Once we go through this stage, again for PAT, I will take a very detailed discussion. And PAT is something which is something which I am very much interested in it. And for the past 10 to 12 years, I have been working on PAT, and we have some very commercial successful applications also, approved projects where we have done PAT and all. So those examples, and in fact those examples will be very easy because whatever work of PAT I have done, they have been already published into some good journals. uh, Two paper, one on NIR and one on the imaging, have been published, and right now I'm writing the project third paper, which is based on Raman technology in PAT, that I am writing. So we have those paper, and recently, last month, I have been to Bangladesh, and I have taken a 2 days workshop I have conducted on QBD and PAT over there. So in that PAT second day, I converted all my papers into a presentation, and I have given a very basic understanding how we go ahead with PAT and all. Same presentations and all, because obviously these are in the public domain, and they have been published with the permission from my organization. So it is very easy for me to speak. So I will dedicate those same presentations I will take, and maybe we'll have a session. In fact, when I did this in Bangladesh, I think it was a very good interactive session, and I got a very good response from the team whom I was training. So I will take this session also. Once we complete the basic course, I will take you through it. Then monitoring and analysis, we have a very dedicated session for this also. Post-marketing surveillance, this is again very important, might be as a R&D scientist, you might not think about it, but as you start growing in your department, and once you reach the position of a head of R&D and all, na, this becomes important for you also, and this is a very important slide for the organization, for the quality, for the production, for every department. This is a very important slide. At R&D, if you are at a junior level, this might not be important, but when you reach a head stage, because whatever you do in R&D, it comes as a post-marketing surveillance once your product is transferred. So the framework is that the strategic process is purposes that continuous manufacturing of product safety, efficacy, and quality to protect public health. So once you start a product, once it is in a market, you have to keep an eye on the product, how the product is going, whether there are complaints or no complaints, whether OOT is coming, OS is coming, and all these details you have to always keep an eye to ensure that post marketing also your product is running smoothly. So in surveillance scope, as I told you, safety, quality, and efficacy are very important. So usually this is done mostly by the marketing team. They are in touch with the physicians and the pharmacists, and they go get the required information. And if there is anything, or even sometimes patients come up with very basic questions and all, and we have to answer with this question. For example, there was a case where we were giving metformin Oros tablets, and the tablet was, the ghost matrix was coming out as it is in the stools. So there were 4 to 5 patients in the US who has put this complaint that your medicine is not disintegrating. In lay words, they have put, and that's why we are not getting the desired effect. And then we have to explain the complete mechanism of osmotic. So this becomes very important for us. Then regulatory obligations, of course, the FDA obligations and the global ICH guidelines, we have to keep on monitoring, and monitoring. Then in case of regulatory compliance, FDA reporting 15-day expedited report for serious. If there is any advance event, you have to report it within 15 days. As a R&D head and as a plant head, as a quality head, you have to report it. These events has to be reported even to the relevant FDA body also. And if there is something which you doubt, you have to recall the product. This is something very important. Then annual reports with cumulative safety data. This is taken care mostly by the QA, but you have to keep on doing this. Field alert. So field alert is nothing but recalling the batches or informing the field that there is some problem with the product. Long back, I was working with USV, and there has been, because I think it was way back in 2006 or 2007, there was a mix-up between two strength of metformin glimepiride combination. So metformin strength was same 500 mg, glimepiride was 1 mg and 2 mg, and there was a mix up in the 1 mg strength. Some 2 mg tablets were found, and this was found by the patient, and then the complaint was raised, and we had to recall almost 15 to 17 batches back from the US to India. This is something very critical. So you have to always take care about these things. Then pharmacovigilance, not that important for us, but again, someone who is into pharmacovigilance and someone who is into labeling and someone who is into the health sector and all, for them, division of a pharma company, they have to take care of this. Quality monitoring, of course, this is monitored by QA, but not only by QA, your supply chain also plays an important part in this. Any complaint needs to have a systematic investigation. Once the systematic investigation is done, you have to have a root cause analysis and trend analysis, and this has to be presented to the FDA as and when required. Stability, whatever. Suppose because when you manufacture a product, you give us 6 months 4075 data on which basis you get the approval, but you have to monitor the long-term stability till that time you are giving the shelf life. In fact, if you are giving a shelf life of 24 months, you have to monitor your 36 month stability also. And if at all, if your shelf life is 24 hours, and if in 18 months some OS is coming and all, then you have to inform the FDA, and if required, you have to change your shelf life also. This is something very important. And then risk management. So in risk management, again, we will have a when we do the QBD, this risk assessment FMEA mode. Again, I'm very much interested in discussing this. Kappa also I will discuss. Again, while working in my current organization, I have been working very closely with the plant people also, and in our organization, whatever kappa is built, it is always rooted through R&D. So in my organization, if anyone is working in R&D, he will know a lot of details about kappa, and also I will discuss this kappa section also. Kappa is nothing but corrective action and preventive action. So if there is any failure, OOT, OS, how to address, how to have a phase one investigation, how to have a phase two investigation, how to have a phase three investigation. We will discuss this in detail, and then how to write a corrective action and how to write a preventive action. So in my organization, we know this very better because you are aware that we went through a lot of problems with the US FDA. So for that purpose, we have got lot of trainings on Kappa, and even now, not even for the US, but for the UK market, EMEA market, ROW market, we have a very full-fledged Kappa plan, properly designed. So anyone who works in my organization, they are very thoroughly having idea of Kappa. As an R&D scientist, it is always important to understand what is your kappa. Just you cannot leave it on the production and the QA person, because you know the technical things and you are the person who should know in and out of the products. So we will come to a stage where we will discuss a detailed kappa section also. as and when we reach that stage. And I have a lot of material related with kappa, because since when we were facing problem with the USFD and all, our organization has hired Mr. Lackman for the quality perspective, and I had got the opportunity to get trained under him, and he has shared a lot of data. Of course, that data is too used data, and there are too many folders. Will take time for me to get the relevant data. But when we reach that stage, I will definitely take a very proper session on Kappa also. Then crisis management, how to recall procedure, communication plan, and regulatory. This is mostly the work of regulatory and QA team, but still you should know. And continuous improvement, mind it well. Continuous improvement is always required. You just cannot leave the product on the plant. R&D has to continuously improve and upgrade the product as per the recent guidelines and keep updating the FDA. So now with this slide, we'll move to the next slide. I think we are facing a lot of internet problem in my city today, because it's been raining heavily since morning. So future trends and innovation. So innovation driven generic development. So now the trend is coming about AI also. Then you have to be digital, you have to precise, you have to be sustainable. So in my organization, again, we are having a very AI driven focus R&D approach. So we will take this, but again, right now it is not important, but we have important other things to discuss, but we will definitely discuss with it. So with AI and all, now the claim is that 50% faster timelines via AI. So when we complete everything, I will again share some practical examples of how with AI we have managed to reduce timeline. For example, now very basic thing in my organization, the APQR document, which is usually prepared by the QA or the CQA, we have applied AI to it to format, and we have connected it with all the BMRs folders and all and all. So a typical APQR which takes almost 10 to 12 days to making in our organization, we are making in 5 minutes. We'll discuss this in detail when we go there. So AI and machine learning, so predictive modeling and formulation is important thing. Continuous manufacturing, end-to-end integration. Again, there are a lot of machines. GIA is the company who is into full-fledged continuous manufacturing in tablet process. We will discuss with detail. I have in fact visited this GIA facility, and I also have information about continuous manufacturing which I receive from them. So that we will discuss. Then the big data insights and digital twins we will do. 3D printing, actually, I don't have much of knowledge and all, but I am also trying to learn. The day I learn it, I will give a presentation on that also. So regulatory, the FDA's streamlined reviews and digital submission already we are doing for ANDA and all the markets. Then the advanced manufacturing framework for continuous process, this is also required. Then the harmonization, global convergence. If you look at my LinkedIn posts 7 to 8 months back, you will get a lot of information about harmonization. We'll discuss this also when we go there. Then in case of R&D frontiers, the new technologies coming, that is the nanotechnology and co-crystal technology. Then the computational technology, that is the PBPK modeling and molecular modeling. Of course, I'm not much involved in this, but I have a basic idea. Green chemistry, again, an important part. And then the personalization, precision dosing also we need to discuss. Mm, complex generics and all we will discuss. Biosimilar, I also don't have much of idea, but again, I'm in a process of learning. We will discuss this as and when it comes. So with this, I end the presentation, and just give me 2 minutes. I have some urgent call, and then we will start with the question and answer session. Just give me 2 minutes. There is some urgent call. I will mute myself for a minute. Sorry for the gap. So I think we have completed the session. I will stop this sharing and maybe you can put the questions and answer and then we will discuss. So let me stop the sharing. I think sharing is already stopped. Uh, so let me go through the message sections. So is there any message or anything you would like to know? Is there anything, any questions? And there are 10 minutes. And if there are no questions, then in that case, maybe tomorrow I will start with a new example of albendazole. There is a presentation. We will discuss how we have put the presentation, how we go ahead with a new formulation development and what things we look into. So no questions? Sure. Uh, are you all able to hear me? Hello? Hello? Uh, I'm not clear whether I'm online or not. Maybe if I'm not online or if there is any issue, maybe we can discuss tomorrow also. Okay. There is a question from Satish. As you said, like we cannot get data on bias trend for EMA. Is it better to write control correspondence for every product? Of course, it is better. They charge high. So the preferable mode is that your regulatory, they do some survey in the market. Based on that, they propose the bio strength and all. So if they propose, they come up with a certain amount of confidence and experience, then they can go. If they have a doubt, or if you have a doubt, it is always better to do a control correspondence. In fact, even for the bio studies also, means what type of study needs to be done, whether a fasting study is required or fed study. Because if you go to US FDA website, you get a very clear idea that you have to do either fasting study, fed study on which strength. In this case, even for that also, you don't have which type of study needs to be done. So it is always better to communicate. I think it charges around 2.5 lakh rupees, but it is always, it saves a lot of time. So if your organization, if your regulatory person is smart enough and he has a ample of experience, then he can guide you. I will put one detailed example, or there is a, I have a Word document on how to select a innovator, and yeah, in case of ROW market, I'll put that. But maybe we can ask someone from regulatory also to explain. And if your regulatory person is smart enough, he will decide the innovator. But if it is not that case, it is always better to do a control correspondence. Then there is a question from what is the timeline for CC? So when in case of US FDA, It is a very clear timing. You take the appointment and they get back to you within 15 to 20 days. In case of MHR and EMEA, they take 2 to 3 months to give you the time, and once you submit the query and all, within 15 days they respond. But getting the slot is somewhere very difficult with the UK MHRA. There is no defined time, but it takes 2 to 3 months. Ali Akbar, can we check IIG limit of excipient? We do. We, even though IIG limit is not mentioned for other market, we check for UK, Europe, Brazil. We try to follow the US limit. When we are developing a formulation which is common for multiple markets, so anyway for the US part you have to comply. So it gets complied by in the UK and Europe also. And even if the formulation is meant only for UK or Europe market, is still always advisable to have be within IIG limit. Otherwise, you get query and you have to keep on answering it. So, any more questions? If there are no questions, then I will log out. Okay, there is one more question. As you said, for USA market, we have exclusivity. For EMA market, we have that. I'm not getting this question. Who is this? Uh, Satish, I'm not understanding this question. For US market, we have exclusivity. For EMA, we have that. I'm not understanding what you are asking. Exclusivity as in when, yeah, we have, we have, we have. Like 180 days exclusivity. Of course, it depends upon the IP dynamics there. And similar to the US market, we have it in EMA and UK also, depending upon the patent scenario and IP scenario. Your IP person will decide and all, but we do get that exclusivity. And it all depends upon the type of molecule and the type of scenario, patent scenario. Any further questions? I think that's all for today. So maybe, uh, okay, one more comment. Okay, thank you. Welcome, Akbar. Uh, Satish, thank you. Welcome, Satish. So tomorrow, maybe night time, I have selected this time. I think many people have contacted me and said that it is not possible because they are working and all. But unfortunately, tonight I have a family function and I didn't want to skip for 2 weeks. That's why I took this session. Tomorrow is Sunday, and tomorrow I don't have any major plans. So maybe tomorrow at night time, maybe 7:30 or 8, I will put the requirement or the event schedule tonight itself, and we will start with the albendazole presentation. I think in that case, that's all. So I'll end this meeting and we'll meet tomorrow. Thank you. Thank you for your time. Thanks, everyone.